Now let us start the main process of translation. The first step is the initiation. We talked in the previous section that there will be an attachment of ribosomes at the place where the translation is occurring. To understand this, let us look at the structure of ribosomes in the eukaryotic cells. There are two subunits in ribosomes. The first one is the 60S and the second one is 40S. The bigger unit has three parts. Region A, which is the aminoacyl site. Region P, which is peptidyl site. And region E, which is the exit site. Remember it with a mnemonic APE, which will be A for aminoacyl, P for peptidyl, and E for exit. The transfer RNA will be reading from the codon, basically from the P region of the ribosomes. So, what are the steps of initiation? Let us have a look. First of all, look here that this is the messenger RNA. Now take an example of any code, say AUG. Do you remember what AUG codes? It is methionine. Okay, so this is the triplet sequence. Now what happens to it is that there will be an attachment of eukaryotic initiation factor 4 at the 5' end of messenger RNA. These initiation factors will allow starting the process, and there will be an attachment of a small subunit of the ribosome at the lower end of the messenger RNA. Now what will the larger subunit do? The larger subunit will have transfer RNA and it will start creating an anticodon for AUG, which will be UAC. Now with the AUG sequence, there will be an attachment of one more factor, which is eukaryotic initiation factor 2. This eukaryotic initiation factor 2 also has GTP. The initiation factor will break down the GTP and convert it into GDP and will release an inorganic phosphate with energy. Now this energy will be released that would help bind the methionine molecule to the end of transfer RNA. When the binding is completed, the initiation factors and GDP will be released and the complex of methionine will be formed. Still confused? Let us quickly revise the process again. First, there will be eukaryotic initiation factor 4. It will stick to the 5 ends and will allow the 40S subunit of the ribosome to attach in the AUG sequence. Then there will be an attachment of eukaryotic factor 2. It will stick to the AUG sequence and have GTP for energy. When GTP will be broken down, it will form GDP and will release energy. This energy will produce methionine molecules at transfer RNA inside the 60S subunit of ribosomes. And what will be the location of the transfer RNA? Yes, it will be in the P region. After initiation, the initiation factors and GDP will be released from the complex. Simple as that. Let us move towards the elongation process now.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.